So, another story that's related to protein structure and actually protein unfolding has to do with the dilemma with gluten. So, if you don't know what gluten is a protein that is found in wheat. So, to sort of understand where this dilemma stems from, we fundamentally have to think about what happens when we consume any protein in our diet and we've digested it. So, all proteins start out as a folded protein. Going to move this down just a little bit here, give myself some room on the top part here. So we start with a folded protein, and the first thing you encounter when you digest something is your stomach, right? And that stomach has a lowered pH, and we fundamentally are going to denature our protein. So here we have a denatured protein. And reminding you what it means to denature a protein is to unfold it, which means we've eliminated any quaternary structure, if it was a protein that had quaternary structure, eliminated tertiary structure, eliminated the interaction of those side chains with each other, and then fundamentally also eliminated secondary structural elements. So eliminated that hydrogen bonding that allowed us to form those alpha helices and those beta strands interacting to form beta sheets. So once it leaves the stomach, then it's going to spend some time in the small intestine. One of the things we've talked about is the increased pH in the small intestine. Now, it's not drastic, but it's about pH 8. It's going to activate enzymes that we have there, and that is going to degrade the protein. So we're going to degrade the protein into smaller peptides and, importantly, individual amino acids. So this is a degraded protein. So two terms that I need you to know the difference of is what it means to denature a protein, which means to unfold it. We're eliminating secondary, tertiary, and quaternary structure. But a degraded protein means we are actually breaking those peptide bonds that hold those amino acids together. And so we are eliminating primary structure by hydrolyzing that peptide bond. So we are going to eliminate primary structure by hydrolyzing that peptide bond. And this is important because we are supposed to be breaking down proteins into individual amino acids because these individual amino acids are supposed to exit the GI tract. And your GI tract is only one cell thick. There is only one cell that separates you from the outside environment. Remember, from the start to the end of your GI tract, it's basically a tube that runs within you, and it's the external environment that is running all through you. But again, it's important that these amino acids are allowed to exit the GI tract because these are monomer building blocks. We'll talk a lot more about this next semester, but the purpose of the consumption of proteins is generally to obtain the amino acid building blocks to either make new proteins or to make amino acid derivatives, things that we need to build to have our body run. So that's sort of the normal sort of course of action. And the idea is that only monomers exit the GI tract. So here is sort of the dilemma, that if only monomers are supposed to exit the GI tract, okay, if a peptide could get out into the bloodstream, this can be problematic. And the reason is because peptides that are found in your bloodstream generally activate your immune system. And that, again, can be a problem. So highlighting that the normal sequence of events is that you don't have peptides exiting out of your GI tract. So why or when would you have pieces of protein sort of exiting out of your GI tract? Well, that happens when you have something called leaky gut. Okay, so you have this one cell thick junction that is your small intestine, and these cells are supposed to be nice and tight against one another. Okay, but if that integrity between that cell junction is broken, you can have pieces that are larger than are supposed to get out. So leaky gut really is the weakening of the junction between these cells. So weakening of the junction between your cells. And I'm going to highlight here when we talk about these cells, these are the one cell thick cells that comprise your small intestine in your GI tract. And you might say, well, what could cause that? There is a protein called zonulin that is going to stimulate sort of the release of these tight junctions. And you might say, that sounds really weird. Like, why would your body want to do that? Well, sometimes your immune system might want to have access to the outside environment. You know, if you've got an intestinal infection or something like that, it's important that your body sort of knows what that invading agent is so it can develop, an immune response towards it. But the problem is if you've got a situation that creates a more permanent leaky gut situation, and you have peptides from things that you've consumed, like the breakdown peptides from gluten. Here's part of the problem is that gluten is broken down into smaller pieces that often can't be broken down further into their amino acids, so you're stuck with these peptides. And if you have sort of a leaky gut situation that allows them to get out into your bloodstream, it can hyperactivate sort of your immune system, and for some people can cause a problem. So we're going to revisit gluten a little bit later, but this was sort of an important topic just to talk about this concept of unfolding proteins. So both denaturing proteins and then degrading them, and understanding those differences and how they map onto what we already learned about with the hierarchy of protein structure.